So how do the genes, those specific sequences of DNA, how do they work? How do we code them to make proteins? Well, just to give you a background, genes are going to be a segment of DNA sequences, those nitrogenous base sequences, that code for the production of RNA that will then encode for a protein. So we take a gene is basically a sequence of bases that are then going to be interpreted or transcribed into an RNA set of sequences, and then those sequences are then translated into proteins. There's only about 20,000 genes in all of that genetic code that we have, so it's only about 2% of all the DNA we have. And this is genes that are coding for functional proteins. There is genetic material, that other 98% of DNA is used for other things, but right now we're just focusing on genes, which is usually the traits that we think about: hair color, eye color, things like that. So ultimately, the protein that's come from the RNA, that comes from the DNA, is an amino acid sequence. And so ultimately, that amino acid sequence of that protein is first determined by the DNA. Again, there are 46 chromosomes in 2 sets of 23. You get 1 from each parent. In protein synthesis, just to kind of overview, you have DNA with the original recipe, so to speak, and that will then be transcribed into mRNA, which is then processed and released into the cytoplasm. The mRNA is then converted or translated into protein. So the important part is that every 3 nucleotides of DNA is going to code for a 3 base sequence in mRNA or RNA. This is called a codon. And then every codon codes for an amino acid. And it's these amino acids together that are going to give us our polypeptide, or our full protein, okay? So we can make a lot of protein from just 20 amino acids. We only have 20 amino acids. And those 20 amino acids are encoded out of a series of 3 base sequences in nucleotides and an mRNA of just 4 nucleotides: A, T, C, G in the case of DNA, and A, U, C, G in the case of RNA. So we end up with a total of 64 possible combinations or codons to represent 20 amino acids. In protein synthesis, again, just to kind of review, we've got DNA that is going to be transcribed, which is transcription. It's going to form messenger RNA. That's supposed to be an M. Messenger RNA is then translated into our protein or our polypeptide. Okay? And again, this transcription is occurring within the nucleus. This represents the nucleus here. That mRNA will then leave the nucleus, go out into the cytoplasm, and ribosomes will work on that mRNA to translate it into protein. So DNA acts as the master blueprint or the recipe for protein synthesis. The gene is a sequence of DNA nucleotides with different nitrogenous bases. And each 3 code for 1 protein ultimately, but the segment of DNA with a certain sequence will give us a protein. And it's triplets of these nucleotide bases that form our genetic library. Each triplet is going to code for an amino acid. And we have 20 possible amino acids.